Hello, everybody. I'm Beth McDougall, and I'm here today with William Brown. And we had an excellent chat yesterday with Nassim Haramein about what they're doing with water. And so today we're on location at Taurus Tech. This is Nassim and William's lab, and we'll have an opportunity to speak more specifically about the technologies they're using. And how they're enhancing water, and then what kind of applications they have for the enhanced water. So William, tell us a little bit about what you're doing and where we're seeing you right now.
Well, yeah, thanks for speaking with me again. So yesterday we were discussing, our discussion got into some topics, including the morphogenic field. So me and Nassim's work, we've described the physics and some of the mechanisms that underlie the morphogenic field, and we call that the unified space memory network. And space memory, and that we're describing an an information dynamic, which is information exchange, feed forward, feedback exchange, and memory properties of space itself that involve the quantum structure of space. This
Mhm.
is a quantum gravitational description.
Yeah, and I think what was cool that came out of our discussion yesterday is that space is highly organized. And part of it's almost like space itself is structured. And that's part of what allows it to hold and retain information, like just like water can hold and retain information.
Yeah, and literally everything is in space.
Hmm.
So it is the reservoir of the information, the blueprint of everything.
It's the Akashic records.
Yeah, yeah,
Yeah, yeah.
yeah. And when you get into some of the quantum physics of space, you see that this is the medium by which everything is connected
Hmm.
and entangled. So a lot of our theoretical work is in looking at how physical systems in general in the universe couple to that field, and that is the cause of formative causation,
Hmm.
which is a term coined by Rupert Sheldrake.
Yep.
So we've got a good theoretical foundation for how that's occurring. Now we've taken that and applied it in some of our technologies. So what kind of systems can you use to enhance or optimize the coupling of physical systems to that morphogenic field, that information structure of space
Hmm.
that is informing things to have their kind of optimal configuration, their optimal dynamic.
The formation of matter. It's like the
Uh,
information, the data set, the information, which what I would see as like almost like a geometrical field type of information set that is kind of directing the flow of the life force into the formation of matter.
yeah, and you know that is one of the missing links on how you get from a universe that is a whole bunch of, let's say, non-living matter undergoing this evolution, and you get living forms out of that,
Mhm.
biogenesis.
Mhm.
So this dynamic is at the root of life.
Right.
And so, what we see as health and vitality in the biological system, to a large extent, that the underlying dynamic that is the coupling with this morphogenic field.
Yeah. Yep.
And, and so if you've got a technology that can augment that process, enhance that process, what you would expect to see is increases in the vitality of the living system.
Oh my gosh. Absolutely. Yeah. And so just because we are talking on International Water Day, talk about how water is interacting with this transfer of information from the unified field into the physical form.
So pretty much every biological activity that's occurring in the cell, among the cells, in the tissues of the body, it's all being regulated and orchestrated by water
Yeah.
from start to finish,
Yeah.
essentially.
Mhm.
So the way that the genes in your DNA are being read and transcribed and translated into proteins that are the functional units of the cell that drive metabolism, that drive cell proliferation and division, rejuvenation processes. Uh, that initial most step is being intermediated via water. So water forms what's called a chiral superstructure along the DNA.
Mhm.
So
It's the spiraling array of structured water around the DNA, correct?
yeah, yeah,
Mhm.
yeah, yeah. And you know, it's chiral, in that it has a very specific orientation in regards to spin. So you know, it's a
Ah,
helical array.
yeah,
So you know, it can spin like in a right-handed dynamic or a left-handed dynamic.
yeah.
And you know, this kind of spin selectivity is hugely important in the biological system because you know, like all DNA has a like left-handed spin.
Uh-huh.
Or I'm sorry, all DNA has a
Oh,
right-handed spin.
right-handed spin, I
Yeah,
think.
right-handed
Yeah,
spin.
yeah.
All amino acids have a left-handed spin.
Okay.
You know, so this chirality is very important. Water does as well. It'll form a handedness on its a
Yes.
spin orientation along the DNA. And now, if you think about it, your DNA is a double helix
Right.
structure. So the base pairs are inside of this helix. So
Right.
you've got the phosphate backbone,
Hmm.
and all the information is on the inside.
Right.
So you've got these transcription factors, polymerases that are going to come and read the DNA, read that. How do they know where the gene is?
Yeah.
Well, you've got 3 billion base pairs, and 1% of that is open reading frames, genes.
Hmm.
So where do
Only
they?
1%. Yeah.
So where do they know to read the gene to start transcribing or start replicating? In fact, what is happening is these proteins, as they're going along the DNA, they're reading the water structure.
Gosh.
Yeah, the water structure is telling the proteins what the sequence of DNA is inside the double helix. And that's that chiral superstructure. So they go along reading the water. Water tells them, okay, here's a transcription start site for a gene.
Okay, so I have a question.
Uh,
So is the water telling the enzymes that by virtue of retaining the information in the coherent domain of the water, is it vibrating in concert with the base pairs, and it's a vibratory communication? Like how do you envision that communication is occurring?
yeah, so each one of the nucleotides um, has a differential effect on the um, electronic shell
Mhm.
the electronic dipole moment of the water molecule.
Okay.
So the water molecule, so I think in our discussion yesterday, we were talking about how it's a partial dipole moment.
Mhm.
And we were explaining what exactly a dipole is.
Mhm.
It's something that, in this case, reacts to electrical charge,
Mhm.
whether positive or negative. So that electric dipole of the water responds differently to each base pair of DNA,
Okay.
whether it's A, G, C, or T.
Mhm.
And so it's going to have a slightly different
Almost
structural
configuration.
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 Exactly, yeah.
Yeah.
And, at the quantum level, this is uh, closely associated with the quantum vibration of
Mhm.
the molecule. So it is vibrational,
Yeah.
rotational.
Yeah.
All those are kind of coupled together, and that's the information that is being read.
Mhm.
So it is like a resonance exchange of
Right.
information.
So I love this because, we alluded yesterday to how water influences which genes are being expressed at any
Mhm,
given moment.
mhm.
And you've just really explained how it's doing that.
Yeah.
And so, just for our listeners today, yesterday we were talking about how we are 99% water by number of molecules. And there's almost not a place in the body where water isn't. And water molecules are so tiny that they intercalate in the inner parts of other molecules, and all around them, forming kind of sheaths of organized, like structured water all around proteins, and as we just discussed, spiraling arrays of structured water around our spiraling DNA. And so water is instrumental for determining, or helping to inform enzymes of where to go and what to do when they get there, with in terms of like replicating genes, or expressing genes, transcribing them, excuse me, and also affecting the way the proteins work. So it's just so exciting. Um, Now, today, one of the things we want to talk about is what you're doing in the lab to enhance water that either we drink or water that is being used in agriculture. Um, and I love what you were saying before, William. Maybe you could touch on it again, where when you're enhancing the coupling between this kind of this information field, let's call it the morphogenic field, and a substance itself, let's say our biology or a plant, um, when you enhance that coupling, you're going to have greater vitality because you'll have a you'll the physiology or the biology will be better informed, and energy will flow through in a more organized way into the system. So tell us what you're doing with water and what you're seeing, like what kind of results.
Yeah. So as you stated there, if you can optimize the coupling of uh, the biological system. So, like for instance, that DNA water superstructure and the information exchange occurring between it and the protein. If you can optimize that information exchange very easily, you can draw a direct link to how that's going to benefit the physiology of the cell, the cellular system, the tissue, the body.
Mm-hmm.
Um, and in every step of the way, there's that water and how it's structured. So if it has an optimal structure, it is coupling to the uh, information dynamic of the quantum field of space uh, to the optimal degree. And that's going to translate into the uh, optimal healthy state of the body.
Mhm.
Uh,
Mhm.
in most instances where you have a disease state, a malfunction of the cell, what is occurring is that there is a degree of decoupling of that
Mhm.
information exchange occurring. And so
Right.
by maintaining the coherent structure of the water vitality, to a
Mhm.
very significant degree, you can maintain the health of the system of the cell. And so we have developed technologies that are going to augment that optimal coupling, that are going to benefit that, and increase the vitality of the system.
Mhm.
So one of our technologies is the Advanced Resonance Kinetics crystal, the ARC crystal.
Love
Uh,
it.
so this is a 8 matrix
Oh.
design. And so what you have here is a modular arrangement of these ARC crystals. So this
Yep.
is a crystalline, a crystal oscillator. And that's
And
another thing we were talking about quite a bit yesterday
yeah.
is, our bodies are crystalline oscillators,
Mhm.
biocrystalline oscillators. Um, the water in the cell in the body is in a liquid crystalline state. Gerald Pollack's work has shown this very extensively.
Yeah.
Um, Even, the proteins, they form via interaction with the water. And those proteins, you can crystallize those.
Yeah.
The way that we know the structure and configuration of proteins in the cell is via
Crystallography.
crystallography.
Yeah.
So you literally are like a biocrystalline oscillator. So
Yeah.
here we have a crystalline oscillator to couple with your biocrystalline oscillator.
And with the fundamental structure or geometry of the unified field.
Mm, exactly. Yeah. Yeah. So
Mhm.
this is specifically engineered for maximum coupling with the uh, energetic oscillation of space,
Mhm.
at the Planck scale, at the Planck field.
Right.
So
Well, at every scale.
at every scale,
Yeah.
to bring that information reservoir at the Planck scale, each harmonic level,
Yes.
into the biological scale.
Biology. Yeah.
Uh-huh,
So
uh-huh.
it's like it's enhancing kind of the communication at all layers of the fractal.
Exactly.
Love
Yeah.
it.
Yeah.
Yeah. So show us the action with water. Like, so what do you, so how does this interact with water?
Well, so um, just a little bit more on the preparation of these
Yeah.
units,
Okay.
because I've got some images I can share.
Okay.
Okay, and so we have our arc crystal, and its dimensions are very specifically engineered, so that we achieve an optimal intrinsic resonance of the crystal oscillator.
Can I
So
just say something about that? Because I
yeah,
think just so everyone in our audience understands, it's like, it's almost like two key of C tuning forks,
uh-huh,
right? So the resonance we're speaking of is almost the same as harmonic resonance in music, where if you ring a key of C tuning fork, another one in the room is going to ring, even if it's a different size, but it's the same shape, it's in resonance.
and that's just the perfect analogy for what we're doing.
Mhm.
I'm not even sure if analogy is the right word because it's almost exactly
Yeah, I
what we're
know.
doing.
It is
Yeah,
what we're doing. And it
yeah,
is
yeah,
harmonic and it is
yeah.
musical and it is all of that. Okay.
Because we have what's called the harmonic flux resonator. And the way that this is tuned is with notes.
Wow.
If you saw the uh, operations desk for like controlling the various parameters of the operation of this electromagnetic resonance generator, we have a keyboard,
Cool.
and you tune it with actual notes. It's all resonance, sympathetic resonance, sympathetic energy resonance transfer.
Yeah.
And so each one of the arc crystals are placed in the field of this harmonic flux resonator, and it's going to ring the crystals, just like that tuning fork. You strike it. If it's at the right harmonic, if it's at the right note, it'll cause our crystal oscillators to ring as well at that note.
Vibrate, right?
Uh-huh.
Just oscillate ever so slightly, I
Mhm.
imagine.
Yeah,
Mhm.
yeah. Well, and if you could see them,
Microscopically,
yeah,
it
yeah,
wouldn't be so slight. It'd be like
uh-huh,
pretty dynamic, I imagine.
yeah, yeah.
Yeah.
And the uh, crystals, it's a quartz crystal, pure quartz, so
Yeah.
optical grade, electronics grade quartz.
Mhm.
And so quartz has some very remarkable properties, like it is piezoelectric.
Right.
It has an electromechanical axis.
So I think you should describe what piezoelectric is to the audience.
Yeah, so it's a really interesting property in that so that ringing, that like a tuning fork that's been struck and is vibrating,
Mhm.
when that happens with a crystalline lattice of quartz, it generates an electrical current
And what about photons too? Like,
and
isn't it
photons.
releasing some photons?
Oh yeah, because
Yeah.
if you've got moving electrical currents, you're
Mhm.
generating an electromagnetic field,
Mhm.
and that can get into a lot of other interesting properties of the quartz because it has what are called nitrogen vacancies that can
Uh,
store high
great.
energy electrons and give off photons, exchange photons. Uh,
Can
the,
you can you store data in those vacancies?
Yeah, yeah,
Yeah.
and even more so than that, um, because they're like isolated
Mhm.
chambers, these
Mhm.
electrons can be in quantum superpositions. They can be
Mm.
quantum entangled. So crystals are used to produce quantum entangled photons,
Mhm.
and those vacancies are used for storing qubits,
Uh-huh.
quantum entangled bits of information.
Yep.
So the crystal is a very remarkable technological
I know.
configuration of matter.
Yeah. I mean,
Uh,
it's like, I think people don't fully realize that these beautiful structures that are just found naturally in the ground. And of course, this is an engineered crystal and it's
mhm,
pure and optical grade and all of that.
mhm.
But crystals are the most impressive pieces of technology, and they've been used potentially for a very long time.
Very long time.
um, we won't get into that. But anyway, so now talk about, again, I keep trying to get us back
Water.
to the water because this is water day. So and I know this does some incredible things with water. So
Yeah, so we have a number of devices that we've constructed for coupling, for arranging the arc crystal in such a way that we can directly treat water with it.
mm,
Because you've got that um, oscillatory vibration, that harmonic resonance of the crystal that we're setting up with the HFR. And then that's
and the
producing
HFR is the harmonic resonance.
generator,
Okay.
the harmonic flux resonator.
Flux
Yeah.
resonator, and it's
Uh-huh.
the big technology that you're informing the crystals with, right?
Yeah, yeah, by structuring space-time.
Yes, and then the crystals themselves are influenced by that.
And
Or
so,
they're programmed with that,
yeah,
so
yeah.
to speak?
So, yeah, when they pick up that harmonic resonance from the HFR,
Hmm.
they're now like a little fractal of it. So the
Got
spin
it.
dynamic that we're introducing into the structure of space time is now being carried around by each of the arc crystals. Yeah, yeah,
And then anywhere they are in the world, they're still in resonance with the device, correct?
exactly. Yeah, yeah,
And with all the other ones, and with space time or space memory in general, right?
yeah, yeah, yeah.
Wow. Okay. And so
A-
now,
And so, that's a coherent field that the water responds to. Again, because it's got that partial dipole moment, so
mhm,
it's going to align to
mhm,
that spin structure.
right.
So now, when we uh, put water into the coherent field of interaction of the arc crystal, it's going to have a very um, significant structuring effect on the arc crystal, and it's going to increase the structuring, the formation of those coherent domains of the water.
And so right here, I think what we have here is your crystals arranged in a helical array. Is that
Yeah,
right?
yeah. So,
Yeah. How
um,
many?
yeah. So what we have is we put the crystals
Hmm.
face to face,
Uh-huh.
and they're tetrahedral units.
Yeah.
So when you do that, it forms what's called a tetrahelix,
Hmm.
which has three helical arrays, three helices. And the way that we arrange them, it's actually at the same ratio, turn ratio as DNA.
DNA. Hmm. Cool.
Yeah, yeah. So,
So now it's in resonance with our DNA. Wow.
yeah,
Okay. Very
yeah.
cool.
And so here we can pass water through the device. It's going to vortex. It's going to
Mhm.
spin as it goes along the tetrahelix unit, because as we were just saying, the spin is very important.
Mhm.
Because the water has a very specific spin orientation
Mhm.
around the DNA in that chiral superstructure, and so in this way we can uh, increase the formation of the coherent domains, the structure of the water,
Right.
with this technological treatment.
And and I just want to say to everyone watching is that there are a lot of devices that are going to move water in a spiral. There's a lot of devices that are flow form structuring technologies for water, and that this is that, but then it's doing so much more than that because it's imbuing the water with the information inherent in the crystal that's entangling it or allowing it to resonate more perfectly with the structure of space memory and with the structure just of the unified field, right? I mean, so
Exactly.
kind of say what sets this apart from other flow form structuring devices, just in a nutshell, like if you're gonna
Yeah, yeah.
sum up.
So that flow form uh, assists the water in forming coherent structure, because spin and torque itself is a force that imbues coherence
Mhm.
to structures. Because you can kind of think of it, water that's just sitting in a glass, like on your table. The water molecules have every different kind of orientation. That dipole can be oriented any different way, and kind of a lot of random motion. So not a lot of structure. Even there though, you do have coherent domains that are forming, but the length of time that they're lasting, may not be very long and may not be optimal for physiological purposes. Now, the moment that you introduce a torque, angular momentum, a spin, uh, all those random motions are going to align in a singular direction.
It almost reminds me of a magnet, you know?
Yeah.
You
Oh,
know what I
yeah.
mean?
Yeah.
Like you have individual iron atoms that have like, their magnetic poles are kind of all at random, directions, and then all of a sudden you just like
Yeah.
line them all up, and then it
Yeah.
does work for you.
And, it behaves a lot differently when they're all aligned, right?
Yeah. It has
It's,
like a coordinated resonance, like a
uh-huh.
coordinated activity that can more efficiently extract energy and information from the field.
Yeah, exactly. Yeah. Yeah.
Mhm.
So then when they're all aligned, it's coupled more coherently, more strongly to the field.
Mhm.
And it has very remarkable properties when that occurs. Very much the same with water.
Mhm.
Now, what the arc crystal is doing is that even after that treatment, it's passing along that resonance, that note. It's really harmonic chords that went from the HFR into the crystal,
Mhm.
carrying that coherent field around. It's a spin field. Now the water's carrying that. Because remember, you also have structural resonance occurring here. So these are tetrahedral units.
Mhm.
Um, and at the molecular level, it's silicon tetraoxide. It's tetrally coordinated as well. So you've got the same geometry as water
Yes.
in the
This
quarks.
tetrahedral
Hmm.
geometry.
Hmm. And
And
so
just
you,
for our listeners, like when you have when you have oxygen bound to hydrogen, the way the bond angle is that they kind of come off of the hydrogen at the same angles as a perfect tetrahedron.
I wonder if I had enough room, I could get it. I don't think the camera's going to reach. I've got that schematic up there of
Cool. Well,
the
we can show a picture of it.
okay,
But
okay,
but
yeah, yeah,
anyway, so I think what our listeners are probably really interested in is the results. So tell us like what you've been seeing in your experiments in the lab with plants. There's the coolest one I want to talk about, and what you're seeing in other biological systems.
yeah. So if the technology is helping to structure the water is going to have a more coherent structure when it's taken in by the biological system. You
Mhm.
should see differences in its health,
Mhm.
markers of vitality.
Right.
We've run tests where we give this treated water to plants and see how they do compared to controls
Mhm.
given untreated water. And what we see is that all the markers of vitality are increased in our plant test groups given the treated water.
Yeah, so the plants are growing faster, bigger, they look more robust, they take longer to die. Even
Yeah,
after you cut them, they stay fresh longer. Correct?
yeah, yeah.
All these
So
things.
yeah. We ran a test with tomatoes. We had increased seed germination rate, increased growth rate, earlier onset of production of tomatoes, and a greater yield of tomatoes.
Wow.
We took those tomatoes and just kind of as a corollary, let's see what happens. We put them uh, in around one of our eight matrix cuboctahedral assemblies.
Yep.
So we had four tomatoes from our tetrahelix group, grown group, and then we had our control away, no crystals. Those tomatoes uh, lasted for over a year
After you pick them?
after we picked them. I mean, it's you've got to see it. you've got to see the pictures.
You know what that reminds me of are the
Oh,
experiments where food's put in a pyramid
oh yeah,
and then it lasts indefinitely practically.
yeah,
I mean,
yeah,
like that's kind of similar because you're really that's a resonating body that's enhancing the coupling with space time and
yeah, yeah. And but we were not expecting to see this kind of result. I
wow.
mean, we were anticipating that it would extend the
Hmm.
shelf life, um,
Hmm.
but like a year, over a year, these tomatoes um, retained moisture. The controls, they were like raisins
Uh-huh.
after I think like 3 months.
Huh.
you know, they were raisins. They had mold on them. After 6 months, it was hard to tell that our PGQ group hadn't just been
Picked.
picked. Yeah, and
Gosh.
if the underlying idea behind that is that it's coupling it to the morphogenic field, that
Hmm.
field is helping to hold its form,
Hmm.
and then the water inside those tomatoes are is more highly structured, so it's holding on to itself, holding within the tomatoes greater, so it's not like evaporating as quickly,
Yeah,
deteriorating as quickly.
it's amazing. Okay, so now the other experiment I would love for you to talk about
Uh,
is uh is one that Nassim has shown me pictures of where the there was maybe like grass or wheatgrass or some type of plant like that, and there was a crystal placed in the center of the plot, and then I don't know, if it was watered also with, the structured water through the helical array, but but what ended up happening in the picture, at least, so you can describe this better, is that the grass was kind of like growing in in interesting rings that were illustrating the structure of the unified field. It was like
yeah.
kind of seeing it in real life, in a larger form.
So we had ran a couple of tests, field tests, right? Because the art crystal is setting up this field,
Hmm.
and there's a spin dynamic in the field, and so the idea was, well, what if we plant a field like with wheatgrass?
Hmm.
Can we see like differences in its growth pattern?
Hmm.
And we had done some of this testing really early on in the manufacture and production of these devices, and we would, in those experiments, we would see like like a bell-shaped distribution, where the wheatgrass directly above the arc crystal appeared to grow slightly quicker than that on the periphery.
Hmm.
Now, when we scaled this up, in some of our first experiments, it did look like there was almost like a vortexing dynamic being imbued in the wheatgrass itself. Now, uh, we have done subsequent testing, and uh, we think that some of that first kind of field effect that we were seeing was more just how the grass was growing. So actually, we haven't uh, been able to repeat
Hmm.
some of those test results.
Hmm.
So, it was an interesting effect that we saw, but in some of our later testing, we haven't seen that occurring at the same level we
Hmm.
did in some of those tests. So we're actually not so certain of that effect. It was
Hmm.
interesting what we saw, but it, in some of the other tests we've done, we haven't seen it
Okay.
reproduced. So
Hmm.
it's one of the things that with the experimentation we're doing, we're still in a large extent characterizing a lot of the properties and effects of the art crystal, And with testing, um, sometimes in some of the preliminary tests, you'll see interesting results, and you may or may not see them repeated with additional testing. So,
Yeah.
you,
Well,
you know,
that's science, and
yeah,
you know,
yeah.
but any other interesting experiments you want to share? Any other experimental results that you guys have replicated and feel confident about?
Yeah. So, um, our plant testing where we see increases in the growth rate is something we've seen repeatedly. So uh, there's little doubt that there's definitely an effect happening there. Um,
And
some other,
it seemed like there was effect with one crystal, there was more effect with more, and that's kind of how you settled on the helical array with
Mhm.
six. Is there a point past which you would start to see a downturn in the benefit, or not?
No, no. So, this is the, 8 units brought together. You can bring 8 of these together into a 64.
Hmm.
And it looks like the effects are nonlinear, in that you get almost like a geometric increase in the effects with increasing modular arrangements
Oh,
of the
wow.
units. Yeah, yeah. So, um,
So the only limitation is probably cost on that,
yeah,
you know,
yeah, yeah.
practicality, because it's very
Uh-huh.
expensive to cut those crystals. And
Yeah,
yeah, yeah,
64 is a lot of crystals.
yeah, yeah.
Yeah, yeah. But so some of the other interesting effects we've seen are direct measurements on water. So during uh, testing with it, repeatedly I've observed what looks like greater energy dynamics of the water.
And how are you observing that?
Actually, in one of the first tests I ever did with Nassim with one of these, um, it was a real simple test. We were just gonna check it out, let's see what happens, kind of thing. Um, we got a whole bunch of lima beans, and put them in a arc crystal test group and a control group, in between sheets of paper towels. You wet the paper towels and you let the seeds germinate, and we can count, okay, how many seeds germinated in our crystal group, how many in the control. Then like measure the stalk length growth, the root growth in each group. And one of the first things that I observed in this testing was that our arc crystal group, the paper towels kept drying out much more quickly than the control group. And of course,
Ah,
we're measuring out the water. Each group is 100 milliliters poured over it. So they're getting the same amount of water at the same time. And our crystal group is drying out much quickly. Of course, one of the factors with that is that the plants were growing quicker. So,
so they were using the water.
they were using more water, but I mean, the paper
Hmm.
towel would be bone dry.
Huh.
And so what my suspicion was is that one of the things you have happening is that resonant information exchange. You have a greater energetic flux
Yeah.
in the water in
It's
proximity.
almost like kinetic movement,
Uh-huh. Uh-huh.
right?
Yeah. Yeah.
Hmm.
Yeah.
Hmm.
Yeah. And it's probably a fairly significant amount of increased energetic flux
Yeah.
because the water's more structured. So
Hmm.
it's holding on to itself more tightly.
Hmm.
So it's quite a bit of energy to have an increased evolution of the water.
Oh,
Uh,
yeah.
and so we have measured this directly now, where we've got water with the arc crystal, and we are taking very sensitive measurements of the water temperature and the humidity level in a controlled chamber. And we've got our R group and a control group. And in our measurements, it is showing greater energetic flux in the presence of the arc crystal.
Oh, wow. Are you guys doing anything with Raman spectroscopy? Like any measurements there?
Yes, so we have done measurements with Raman spectroscopy, infrared, fast Fourier transform infrared spectroscopy. And, that's another one where we were expecting to see um, shifts in the absorption spectra of the water that had been treated with the crystal. But actually, the testing we did with that, we didn't see shifts in the absorption spectra.
Mhm.
Um, so that was kind of interesting. We haven't ruled that out just yet because
Mhm.
um, there's a lot of different considerations when taking those kind of measurements.
Yeah,
One of the things is that a lot of the labs we were working with, they don't normally just measure just water.
right.
They use
Something in water.
something in water. Yeah,
Yeah. And I
yeah,
mean, isn't it somewhat dependent on the strength of the laser as well?
yeah. There's a lot of factors, and
Mhm.
also, water has three axes of rotation and vibration, rovatory properties.
Mhm.
And so when we were probing it, we were looking at that bond angle that is 104.5 degrees.
Mhm.
That's what determines the electric dipole. But
Mhm.
that's just one of
Yeah.
your dimensions of, well, I guess two, your dimensions of vibration,
Mhm.
reverberation characteristics. One that we haven't looked at yet with spectroscopic means is the oscillation of the hydrogen um, in what you could call like the x-axis, where it's actually oscillating like away and towards the central oxygen um, atom.
Mhm.
That one's very interesting
Mhm.
because that vibrational characteristic of the water has very significant metabolic effects.
Mm.
So, for instance, when you have deuterium
I was just gonna
water,
ask you about
oh,
this.
yeah,
Okay.
yeah, yeah. So you're familiar with
Mhm.
deuterium water?
Yeah, and just how health is really impacted adversely when your deuterium levels raise in the body.
Yeah,
Yeah.
yeah.
Hmm.
And
So
so, I
first,
mean,
first off, let's just say to everybody out there, so deuterium is would you say it's an isotope of hydrogen?
yeah,
Yeah, so it's a different form of hydrogen, and it has some different characteristics, and oftentimes if you eat a diet that's really heavy in deuterium, which unfortunately is found in vegetables and things like that, and not found in meat and as much, and so if you're a vegetarian, for example, your whole life, sometimes you're going to accumulate deuterium in the body, and it's going to actually affect even energy production in the mitochondria, and potentially have some deleterious effects in the body because it doesn't function in the same way that regular hydrogen functions. So go ahead.
yeah, yeah. So, but why would that be? This gets into some interesting things because deuterium is an isotope of hydrogen. Isotopes have the exact same chemical behavior. So deuterium is chemically exactly the same as hydrogen. And
Right.
if we're talking about
But it's isn't it heavier?
yes, yeah,
It's
yeah,
heavier. So
yeah.
it binds, it takes up those hydrogen binding sites, but then it kind of weighs the molecule down a little bit. Is that
Well,
accurate? Is
well,
that accurate?
not exactly.
Okay.
Because when you're talking about the weight of some of these macromolecules, these biomolecules, an extra neutron is negligible.
Okay. Okay.
It's actually, though, it's the weight of the deuterium in the water that is having the effect. So, chemically, deuterium D2O,
Hmm.
deuterium water, is exactly the same as H2O. So, naively, it should have the same biochemistry. But because of that increased nuclear weight, having that extra neutron, the
Mhm.
uh, deuterium has a much tighter oscillation, a much,
Mm.
you could almost think of it as like a lower frequency vibration on that oxygen central atom in the
Mm.
D2O. And so what that equates to is that that deuterium is much more tightly held by the oxygen,
Yeah.
and it's less readily available to participate in oxidation reduction reactions
Mhm.
in biochemistry.
Mhm.
And
Mhm.
so, like if you look at the, you know, chemical networks in the mitochondria, so much of it is proton and electron exchange,
Yep. Mm.
oxidation reduction reactions. Uh,
Right.
and so if those are slowed down because you have deuterium, because it's more tightly bound to the oxygen because it's heavier, so it's in a closer orbit or vibration,
Mhm.
it can slow down metabolism enough that it has deleterious effects on the cell, on the biochemistry.
Right.
Yeah,
I know. So people are doing things like drinking deuterium depleted water to try to gradually rid their bodies of
yeah, yeah.
deuterium.
Uh,
And you can actually make deuterium depleted water.
mhm.
Are you familiar with that?
No, how to make it?
So you can freeze water, and then the part that freezes first on the top has most of the deuterium in it. And it's not because deuterium is lighter and floats, it just has a different freezing point.
Wow,
And so then you can pull that like sheet of ice off the top, and then you can do it again, pull that off and pull it off until you've kind of depleted the deuterium in the water.
wow, wow. Yeah, yeah. So that's a really good practice because deuterium depleted water is very healthy.
Hmm.
Much more healthy.
It is.
It also gets into some quantum mechanics because if you're looking at the spin state, um, pure hydrogen has a spin of 1/2,
Mm.
which spin 1/2 nuclei are uh, very weakly interacting with external electronic and magnetic fields.
Mm.
Interestingly, what this means is that if you have two spins that become entangled and their spin 1/2, that entanglement is long lasting.
Cool.
Yeah, yeah.
It's almost
So
like match made in heaven.
now, when you have deuterium, it's no longer spin 1/2. It will decohere much more rapidly.
Oh boy.
You
Yeah.
know, so it's actually starts to get into some quantum biology also.
Yeah.
Yeah.
Oh my God. That is an exciting field, quantum biology.
Uh-huh.
Okay. Well, we're a bunch of nerds, so I don't know that everyone agrees, but to me, that sounds like I want to study that. Anyway, okay, so this has been amazing. And any other experimental results? Just a quick pearl what you've learned working with this technology as applied to water?
Well, it really has been very insightful to me on the role and importance of water. And so some of my just most like uh, theoretical interests in biology, like biogenesis, where does life come from, these kind of things. Um, Being able to work with water has actually informed those questions to a large extent. So I've been able to see the importance of water, the central importance, in some of these foundational questions that are really, at the heart of motivating my research. And so, yeah, it's been um, very rewarding in that sense, working with water. And then, our technology, because, if you think about the Earth, it's like the crust, it's like 70% silicate minerals,
Uh-huh.
i.e., quartz. The surface is like 75% water.
Yeah.
It's kind of interesting that when you get quartz and water in close association, you have this thriving biosphere.
Ah,
So it's been kind of cool working with those systems.
oh, that's beautiful, William. Thank you so much for sharing your beautiful work, and your mind is always such a it's just it always astounds me, and I enjoy talking with you, and you just seem so inspired by what you're studying, and it's infectious. So thank you so much for being in the world, and I share that also with you, where water has been a huge teacher for me. So
Well,
good.
thank you, Beth, for
We'll continue to study it together.
yeah, yeah.
Okay, bye. We'll talk again soon.